Hello everyone, Assalamualaikum warahmatullahi wabarakatuh. Welcome to my video. And now I make this video to talk about the journal I have reviewed. Let's get started. Talk about this journal article. Every journal has its own identity. The first is the title. The title is Novel Mutation in Calcium Calmodulin Dependent Protein Kinase 4 CNK4 Gene in Infertile Men. This journal was included in the International Journal of Andrology, Volume 35, page 810 to 818. This article was written by Katri and friends and was published in 2012. And this article was reviewed by me in August 25 this year. And let me show you the cover. Next, the aim of this study is to identify the affection of CMK4 gene mutation in infertile man's sperm. The dependent variable of this study is the mutation of the kind of gene, and the study was occurred because of the recent studies by Boivin et al. 2007 shows that approximately 10% couples face problems in conceiving a child after trying for a year, and according to Machade and Horst in 1997, the infertility of male partner is the cause in approximately 50% of such cases. The subject of this study is blood samples from men attending the Infertility Institute and Research Center in Secunderabad, India, and the Institute of Reproductive Medicine in Kolkata, India. The extent of CMK4 gene in ethnically matched 283 infertile and 268 fertile Indian men. Informed written consent was obtained from all participating individuals and permission to proceed was granted by the Institute Ethical Committee. A total of 283 infertile Indian men who satisfied the above inclusion criteria were included in the study from our repository of nearly 800 samples of infertile men. The method is CMK4 gene knockout. And what does gene knockout means? Let's figure it out. Gene knockout is genetic technique in which an organism is engineered to carry genes that have been made inoperative. Those organisms carrying such genes are known as knockout organisms or simple knockouts. They are used in assigning function to specific gene having a known function that has been sequenced. The first steps of this method is PCR and sequencing. DNA from blood samples was extracted using a protocol described previously. Primers were designed and custom synthesized to amplify all the 11 exons of the CMK4 gene. PCR and sequencing of each sample was performed as described with annealing temperature and using bioinformatics tools such as databases. The second is cloning and sequencing of PCR products carrying heterozygous deletions. Heterozygous insertion deletions or indel mutations were confirmed by cloning by applicants having mutations into the P-MOS blue factor following the manufacturer's instructions. At least 10 positive clones were screened and sequenced as described previously. Third is statistical analysis. Statistic package for social sciences estimate the risk of infertility associated with mutant alleles and genotypes. Haplotype-based association analysis was performed using Link version 1.5 following the owner's instructions. And fourth is population stratification analysis. A total of 47 of the 50 markers passed the quality control filters. Principal components analysis and ANOVA statistic for the difference between infertile and fertile men was performed using AgentSoft software. The last step is in silico analysis. In this step, also using the role of databases, the first in polybrand and SIFT, and also as finder. Polybrand and SIFT predict the functional impact of amino acid changes by considering evolutionary conservation, physicochemical differences, and proximity of the substitution to predicted functional domains and or structural features. As finder, release was used to evaluate the presence of exonic splicing enhancer at the mutation sites. The fertile man-specific intronic mutations were analyzed using human splicing finder. The last but not least is the results. Talk about the results in the studies are: first, sequence analysis of the CMK4 gene showed that 20 SNPs and 5 indels, you can read the detail by yourself, were exclusively found in infertile men. All these mutations were always found in heterozygous condition and no man had simultaneously two mutations. Second, of the total variants in the CMK4 gene, 9 were only observed in infertile men. Next, infertile man-specific mutations can alter or diminish the potential binding sites for the proteins. And the most important from all these results is the regulatory sequence tool predicted that the 3 infertile man-specific mutations diminished the potential intronic splicing enhancer and exonic splicing silencer sites. Analysis of fertile man-specific intronic mutations using HSF tools did not predict any change between reference and mutant sequence. And finally, talk about the strengths and the weakness. The strength of this study is: first, the knockout gene method can identify genetical abnormalities in sperm cell in infertile man. The second, it can reveal the gene mutation in CMK4 gene that generally affects sperm regulation and motility. And how about the weakness? The weakness is: first, the knockout method using SNPs in intron 2 and in intron 5 didn't show significant difference between infertile and fertile men. However, they deviated from the HWE in infertile men. The second, infertile men having mutations in CMK4 did not show any reproductive tract abnormality and their testicular size were within normal range. Okay, that's all my videos. I don't know if it's really educational, but thanks for watching my video. And see you soon. Assalamualaikum warahmatullahi wabarakatuh.